my intent here is to kind of just point folks to resources that are available. There's a lot out there as far as resources for developing a response plan or a management plan for HABs. And so a lot of the information in the slides are hyperlinked. So when you do get a copy, you can follow the links and download. There's templates. There's all kinds of stuff. So this is kind of just a slide with links to general resources for developing management plans or response plans to HABs. US EPA, the first three links, there's quite a bit of resources. So there's templates, there's example plans from systems that have done this. There's a document called Cyanotoxin Management Tools for Public Water Systems. It's kind of what the headquarters calls like a toolbox, but several resources at that link. And then a document kind of explaining recommendations for public water systems to manage cyanotoxins. And there's and I'll show some screenshots from that. In addition to, on the federal level, a lot of states have developed resources. I'm probably most familiar with Ohio just because we work with them and I'm physically located in Ohio, but they have resources for developing what they call HAB general plans for public water systems, and all of their systems that are susceptible to HABs have to develop these. Basically, it's just kind of an outline of, if this is how we're going to monitor, this is what we'll do if we find toxins in the raw water, and then this is our plan for treatment if we do. And so that's also outlined in their public water system response strategy. And so those are two documents that Ohio asks their susceptible water systems to develop. And then finally, bringing it a little closer geographically, Washington DOH has some guidance on making plans for HABs. I'm sure there's others out there. These are just what I'm aware of, and there's plenty here to get folks started that haven't really thought about this yet. So this is a figure taken from that recommendations for public water systems document that I have a link to in the previous slide. And it's just kind of a flow chart to kind of a high level flow chart to get water system operators, system managers, state folks just thinking about like a stepwise approach to thinking about if there's a HAB in the source water, what to do. And so I'm not going to go into all the details here, but you would start kind of with like an evaluation to assess whether your water body is vulnerable, and I would say most operators probably already know to some degree, that if they have algae blooms yearly, or if they're starting to get concerned about it, they're starting to see algae or cyanobacteria, and then thinking about preparation, thinking about monitoring. We had a session on that this morning, and then both raw and finished water, and then what happens whether you detect toxins in either of those places. And then step 5 has and each of these steps has quite a bit of info and resources and stuff within the document. But another big part that I think we all kind of learned through the Toledo and the Salem incidents, where it's just the public communications aspect of HABs. What do you do if you find it? How long do you have to notify public? When should you notify the public? You don't want to create panic when you don't need to, but at the same time you want to be transparent and let folks know what's going on. So I'm just kind of taking some excerpts from some of the state resources that are out there. So the next 2 slides will just be kind of an outline of what Ohio asks their water systems to develop. So their systems have to develop a document, and a lot of times they'll do this in with the assistance of a contractor for especially smaller systems. And I think at least Ohio provided some grant funding for smaller systems to get that help to put these plans together. But they start with an introduction, talk about their current conditions of their raw water sources, their treatment, how their distribution systems function, because this all plays a role in if you do detect toxins in your finished water, how quickly can you flush them out of your distribution system? How long can you survive on treated storage that's already out there that doesn't have the cyanotoxins, et cetera. And then any future conditions. So, if I know a lot of systems conduct, like water resource studies, thinking about future average and peak day demands. So especially if there's, big expansion plan for a system, how are you going to factor HABs into those plans? And then, Ohio asks their systems to talk about what they're doing currently for cyanotoxins. Based on this analysis, what would they do in the future? Proposed management and treatment approaches. Residuals disposal is important. I think Larry and Bill during their treatment presentations talked about sludge handling and your clarifiers. If you're, we showed that the settling process is pretty effective for removing the cyanobacteria cells, but then what happens when they're just sitting in the sludge? That probably needs to be processed more frequently than normal, in case those cells are still living and maybe releasing toxins. And so that's a consideration as well. And then, just a schedule for, when are these proposed management and treatment approaches going to go into effect? So that's what Ohio does. I'll transition now. If you recall from one of the earlier slides, that stepwise approach that US EPA put together, they talk about in the first step, assessing vulnerability of your source. And so this was something that the state of Wyoming and Region 8, EPA Region 8, which has primacy for the drinking water over Wyoming, they had that question, which of my water systems are vulnerable to HABs, and how can we assess that? How can we direct resources to those systems that are going to be facing the greatest challenges? And so I worked on a project with them to do that, to take data from all of their surface water systems and kind of have basically like a risk assessment or a vulnerability assessment for their surface water systems. And so the next couple slides, I'm just going to show the factors that we ended up deciding on to feed into this risk assessment. So we looked at several aspects of a public water system, right? So their source water, how at risk is your source water? That was the first aspect. And so these are the factors that fed into the risk score, if you will, fed into the risk score. These are the factors. So watershed nutrient loading, and I can talk a little bit more detail about where these came from, where this information came from. I think Wyoming here and Region 8 were a little unique in that they had a lot of this information. I'm not, I realize that other folks in states out there that might not have all this info, but this is just something you can consider. And if it has to be less than this, it's still better than nothing. But watershed nutrient loading, nutrient concentration. So I think they had, through a project, they had done some characterizations of their water sources, as far as nitrogen and phosphorus go. Algal biomass, that can be, a lot of the information for algal biomass came from the satellite imagery projects that were conducted throughout the country. Percent cyanobacteria. So those satellite imagery can distinguish between cyanobacteria and regular algae. HAB densities. So if there's like certain concentrations near intakes, that's factored in. And then finally, toxin concentration. So there's on the recreational side, there were a lot of data available to determine the source water risk. What else would feed into a risk assessment? So in addition to your source water, how vulnerable is the plant itself if there is a HAB? So intracellular toxin removal is another factor. And by intracellular toxin removal, we're basically saying, like, how good is the plant at how good would the plant be at removing cyanobacteria cells? And so these are factors we thought would play into that. How good is their coagulation flocculation process, pre-sedimentation? Do they have dissolved air flotation, which has been found to be pretty beneficial to disrupting cyanobacteria, their buoyancy, and rendering them kind of unable to survive? Do they have pre-treatment oxidation? We just saw that, the effect that adding permanganate as a pre-oxidant can have on helping out your treatment process downstream. sludge removal, media filter performance. So just looking at turbidity, I think we had some tiers for different effluent, average effluent turbidity levels coming out of the plant. Does the plant employ membrane filtration? And then their current plant, how close is the plant to their design capacity? And then moving to extracellular toxins. So these are some of the factors we thought would play into a plant's ability to treat extracellular toxins that come through that conventional process. So does the plant have activated carbon, whether it's granular or powder? Do they have membrane treatment that can filter the toxins? And research has shown really only nanofiltration and reverse osmosis have the ability to do that. Advanced oxidation processes, and what's the plant's CT? How are they doing with CT? So all these factors kind of play in. We had a couple more too. So we thought about, do plants recycle water? Are they sending, concentrated toxins back to the front of the plant? Source water treatment? Are they adding, I don't know, copper sulfate or some kind of coagulant in their source water to handle, yearly blooms? How, their raw conditions of their raw water intake, the flexibility of, drawing from different levels, perhaps to find water that's not as impacted by the bloom on the surface. Any enhanced process controls? Are there clarifiers outside? So are they exposed to sunlight where cyanobacteria could grow within the plant? Do they have aeration at the intake? And we talked about aeration previously on the intracellular side, dissolved air flotation, but some have aeration at the intake, which would be beneficial. Do they have alternate sources that aren't impacted by HABs? Do they have like a groundwater source that they can switch over to during a HAB? What kind of cyanobacteria monitoring is the plant doing? And I noticed that factors in there twice. So, but anyway, all of these factors are, were assigned numerical like ranges, and then rated, and so all of these fed into a risk analysis that was developed for all the surface water systems in Wyoming. And so, all of this information that might not be available to folks, at specific water systems or states, but yes, these are some factors to consider if you're looking to assess the vulnerability of your source. So some response plan strategies, and this might be a little bit of an overlap from Bill's presentation, but when thinking about a response plan, you want to have a treatment plan, ready to some degree. And so these are some strategies that could be employed as part of your response plan. So as far as source water protection goes, one thing we found is that a lot of states have pretty siloed programs, between drinking water and recreational, like beach monitoring and stuff. And HABs have kind of required states to coordinate with the recreational folks. There's a lot of data out there that, they've been collecting for years in some states that's not necessarily making it over to the drinking water folks. And so that's, especially in the project with Wyoming, state of Wyoming had primacy over the recreational water, and then EPA has primacy over the drinking water. And so it requires coordination, and you can get some good information from that side of the agencies. Nutrient reduction strategies, that's a big topic, and I realize that there'd be a whole day's worth of training itself. But and then I know Bill mentioned this, avoidance strategies. They're pretty powerful, especially if you don't have a lot of options at your plant to treat. Intake relocation or pulling water from a different depth within a water body that may not be as impacted, or alternative sources. Can you purchase water from a neighboring system? I'm trying to think of some other, groundwater, if you have some backup wells or something like that. So, avoidance strategies can be pretty good if you have storage and you can shut your intakes. That's another one. Reservoir management. So I know some utilities have had success with ultrasonic treatment that basically works by, again, disrupting the cell walls of the cyanobacteria, and it impacts their ability to move up and down in the water column depending on the availability of sunlight. So aeration and mixing, again, that kind of just disrupts their ability to proliferate in a water body. It makes the water less stagnant. Strategic algicide application, and I use that term strategic, that's put in there on purpose, because algicide's kind of a double-edged sword. It can obviously it can kill cyanobacteria and algae, but at the same time, you do have to be concerned about potentially lysing cells and creating a dissolved extracellular toxin problem that's a little bit harder to treat. So I know in Ohio, they have thresholds for applying algicide. So when, if you're a water system that wants to apply an algicide, especially if you're a bloom impacted water body, they say, above a certain level of cyanobacteria concentration to stop adding the algicide so that you don't exacerbate the problem. And then coagulation, some utilities will add like alum to kind of pre-coagulate some of the raw water. So these are all strategies you can take within the reservoir to reduce the problem from even getting to a point where it becomes a problem for your treatment plant. And then treatment optimization, so coordinating with laboratory personnel and contract labs. So having plans in place for, taking samples, whether it's, a contractor that does it, or, having the resources and contacts in place for if, a bloom does come and you need to get samples with a quick turnaround time, having that, those options in place prior to the problem is important. So as far as monitoring, we talked about monitoring from like maybe more of the scientific standpoint, but as far as putting it in your general plan or your HAB response plan, it's important to get that laid out prior to having to react. So in Ohio EPA, systems are required to submit their general plan if total microsystems exceed 1.6 micrograms per liter in the raw water, and that number is the higher level health advisory for all public. So above that, it would be a do not drink or a do not drink order in Ohio. And so you can see the stipulations on that, or when it's detected in any finished water sample. So this is just what Ohio does. I have a link here. This is a hyperlink. So talking about how frequently to monitor depending on the season, and what happens with certain sampling levels, certain sample result levels, and what those triggers, what steps a plant should take if they meet those triggers. Some other things to think about as far as monitoring go for a plan, coordinating with other agencies. I kind of mentioned this, other agencies that might be conducting recreational monitoring on water bodies that also serve as drinking water sources, coordinating with laboratory personnel and contract labs. So having those relationships in place already. And record keeping and trending of data are important. If you recall from my presentation this morning, I showed that the pH and turbidity plots from that plant in El Salvador. And so if a plant was trending that data on a daily basis and they start to see the pH jump up, up a unit, that's an indication that something's happening that's, not normal. So, keeping records of your daily data and actually plotting them out and trending them and looking at the long-term trends is super helpful for planning and responding to HABs. And then establishing thresholds and triggers for taking action. So I included a lot of the resources that are out there. EPA has some resources as to what to do for certain, if you detect if you detect cyanotoxins above 0.3 in your raw water, here's what you should do. If it's this, here's what you should do, et cetera. So that these are all things to think about as if you are going to put a plan together, put something on paper. These are important things to think about. Continuing with the monitoring theme, as I said, regular monitoring is important. Trending the data is important. It's within the plant too that's important. So if you have the resources to conduct plant profiling. Even doing one when you don't have a bloom is helpful to compare for when a bloom does happen, and that can kind of highlight where the plant's having issues treating the harmful algal bloom or the cyanotoxins. So, I covered a lot of this in the monitoring presentation, but, in addition to actually monitoring for cyanotoxins, you can use indicator parameters like pH. We talked about the pigments, chlorophyll A and phycocyanin. Organics can kind of indicate an increase in cyanobacteria, especially if you have a good handle on your baseline. Turbidity, taste and odor. Taste and odor was a big one for that El Salvador plant. Cyanotoxin producing genes. We talked about how Ohio uses that as a kind of a predictor for toxin production. And then, cyanobacteria cell counts. And I think these two bullets are repeats from the last slide. So, and then, you talk about monitoring, you talk about the triggers, the action, what actions you need to take at those triggers. Then you get into treatment optimization. So if you do have a HAB that's coming into your plant, and this was a set of slides adapted from a workshop in Region 10, so I apologize for that. But we did cover a lot of the treatment optimization approaches. Larry and Bill in their 2 presentations covered those. And there's several resources out there for what treatment is effective in what situation. So the US EPA's authored a document called Water Treatment Optimization for Cyanotoxins. It has a lot of the information that Bill and Larry covered this morning. And then Ohio EPA also has a protocol for optimizing treatment. So these are hyperlinked. You can go and download these documents, or if you have any issues, I'm happy to send them to you. So there's plenty of resources out there for optimizing treatment. So applying the optimization tools for harmful algal blooms. Larry, Bill, and I, and Jennifer, we work on what we call the areawide optimization program, and so that program really emphasizes using existing facilities and optimizing them prior to going and buying a brand new GAC filter or a reverse osmosis membrane. Try to get the most mileage you can out of your existing facilities and infrastructure. And Bill mentioned this, focusing on particle removal is usually the first strategy, and this can be part of your HAB response plan too. Usually during a bloom, most of the cyanotoxins are within the cells, majority between 50 and 95% based on the research. And it does depend on the species. Cylindrospermopsis, for example, it's another cyanobacteria that, while Microcystis, most of the cyanotoxins are usually during a bloom, they're usually mostly within the cell. Some other species like Cylindrospermopsis can they release toxins a little bit more just by their biology or whatever the mechanisms are. And so that's where cell identification is pretty helpful, to understand where the toxins might be too, whether they're in the cell or outside of the cell. And then, minimizing pre-oxidation is if you're able to, it's probably the easiest way to avoid release of cyanotoxins. We did see in the previous workshop where adding a pre-oxidant was helpful, but keep in mind again we made the assumption that the toxins were extracellular, and there are situations where that is the case. Larry mentioned the Columbia River in Washington, where those systems were seeing mostly extracellular cyanotoxins from a benthic bloom. So, that's where the sampling, the sample prep, taking samples to understand intra versus extracellular will be beneficial. And then, emphasizing the multiple barrier approach. So if you're doing a good job monitoring, hopefully you have a good idea of how each barrier performs during a HAB. But thinking about each barrier, during that emergency situation is helpful. So what do you, with your source water, what are you going to do with your algicide? When are you going to turn it off to avoid lysing cells? At what point do you change intake levels if you have that capability, or when do you switch to an alternate source if you have that capability? Optimizing your conventional processes, if you have them at your plant, is something to think about prior, to the bloom happening. So get that in your response plan if you decide to make one. And then removal, we talked about absorption and oxidation in the workshop, and some folks in my group made a good point. Although we were finding like higher chlorine requirements for oxidizing the toxins, doing that exercise prior to an emergency is helpful, because then you can think about, you can put in your plan, like, I know I can't add this much chlorine, so I'm going to slow my, sorry about the dog. I can slow my plant flow rate down to achieve that CT, or having that foresight and thinking about what you're going to do with these processes prior is very helpful. So it's just, I've showed this figure before, just the multiple barrier strategy. Taking samples, within the process is helpful. So some proactive steps to prepare for HAB events, and again, this is all in the along the theme of preparing some kind of documentation, or having a plan of action for when a HAB shows up at your intake. So you can run through some what if scenarios. I know that someone had mentioned leading up to this training that Idaho had conducted some tabletop exercises with some systems. So I have a feeling like it was probably something similar to this, where you conduct some what if scenarios. Can you feed pack based on your analyses, like the analyses we did today? How much pack do you think you can feed? And then you can start thinking about, your feed line sizes, your equipment, where are my limitations? Evaluate the necessary CT to oxidize cyanotoxins. So you can kind of play around with that cyanotox tool to evaluate, if I lower my pH, what does my oxidation look like? If I slow my flow rate down and increase my contact time, what does that do for my necessary CT, et cetera? And then look at feasibility and treatability of alternate sources. I know some systems have emergency connections in place with neighboring systems, have emergency groundwater sources that they can use, but understanding how your treatment might need to change if you do go to those sources is also important. Preparing tools for evaluating treatment scenarios. So having on hand, a jar tester, you can test coagulant doses, pack doses, understanding how to do jar testing and having a plan for doing that is, obviously part of a HAB response. Doing oxidant kinetic studies, that's basically like using the cyanotox spreadsheet. And then developing SOPs, guidelines, goals, and policies. This is all part of, developing a HAB response plan. So having a source water monitoring plan in place, and then having those triggers, what you're going to do when you do find something. And then documenting for all folks at the plant, what are the maximum chemical feed capabilities, where are our roadblocks, and then having plans for addressing those prior to the bloom is important. So thinking about planning for short-term needs and long-term needs, I've got several bullets listed here. So consider any necessary system modifications, like our group talked about, during the oxidation workshop, maybe adding some baffling to the clear well to improve the efficiency of the clear well, increasing the baffling factor. But budgeting for equipment and chemicals, if during a HAB that you're going to need a bunch more pack, maybe budget for that during HAB season. Having plans for purchasing equipment, sampling needs, lab capacity, turnaround time for samples, and action triggers. And that's a big one too. If you see a HAB, you take a sample, and you need to send it to a lab in Florida, and the turnaround time is a week, it's good to know that that's going to be the case in an emergency, so that way you can anticipate that lag in sample results, and then take any precautionary steps in the meantime. And then planning for long-term needs. If your source is continuously impacted by HABs, think about alternative sources, emergency connections with neighboring systems, and treatment modifications. I know there's a lake here in Ohio that has year-round HABs, and they have pretty much every treatment technology available, and that's just the way I guess that's just the way that they're approaching the issue, instead of some of these other long-term needs. But It'll be different for every plant and depending on budgets and things. So the last step, so, we've kind of talked about this from like a science and engineering perspective, but, communicating to the public, keeping public confidence in drinking water is obviously important for all of us. And so that's that's something that I think the industry's kind of learned is super important over the last several years, and so there's several resources on that. I'm not a communications person. We have folks at EPA that work more on that end of things, but there's lots of resources, a lot of templates out there. So I'll just mention these, but these are all hyperlinked, so you can go to these websites and download the templates and stuff. This drinking water cyanotoxin toolbox has a lot of graphics that you can post at water bodies, like example social media posts if you're trying to get the word out that there's a bloom. There's a water contamination response resources. So this is a little bit more general set of resources for any sort of water emergency, drinking water emergency, but it can be useful in this situation as well. CDC has something similar, as far as communicating to the public during a drinking water issue. And then Oregon has developed a set of resources and templates as well that likely grew out of that Salem incident. So I suggest folks take a look at those resources. And then finally, this is EPA's quote unquote traffic light approach. So we were talking about, we kind of went through all these different tiers. We talked about monitoring, treatment actions, and communication. And this is just kind of to a very high level, just to kind of sum up everything that's in that EPA's recommendations for public water systems document. And so you can see that it's tiered based on, what you're finding in finished water, and the different steps you take from a communications perspective, a treatment perspective, and then, a monitoring perspective. I won't read all the little boxes. I'll let folks, investigate those on their own time. But so that's it. I don't know what our plan for questions is, Larry, but.
Thank you, Tom. And